My research revolves around statistical genetics. That is, the development of quantitative methodologies for the analysis of high-dimensional genetic data from next-generation sequencing platforms. In particular, I specialize in the microbiome, which is the collection of microorganisms that inhabit the human body. This is an extremely popular field of study at the moment, as changes in the composition of the intestinal microbiome have been shown to be associated with all sorts of diseases. Microbiologists are collecting a great deal of microbial sequencing data. However, there are many statistical challenges in analyzing this kind of data. In my own research, I developed novel statistical techniques for analyzing microbiome data. The most interesting thing about my research is it combines two very hot areas in research currently: microbiome data and data science. There are a huge number of studies out there that are measuring microbiome composition. Similarly, data science, that is, quantitative techniques for analyzing and identifying patterns in data, has exploded in popularity over the last decade, especially with the growth of AI modeling. I really like that my field is able to touch two completely different areas that lots of researchers are talking about currently. The main challenges we have are computational in nature. With the size of the microbiome datasets, in particular the large number of microbial taxa sampled, there are challenges in designing statistical methodology that can handle large datasets in an efficient manner. Additionally, microbiome data are tricky for statistical modeling since they don't have a nice distribution, that being variables that don't have a typical bell-shaped distribution we study in statistics courses. I have been developing novel methodology in dimension reduction, which attempts to take all of the variables in a microbiome dataset and collapses them down into a smaller number of representative variables. This reduced dataset is much more feasible to analyze than the entire dataset. My PhD student, Saurabh Panchasara, has developed a method called zero-inflated factor analysis, logistic skew normal multinomial model, that performs this dimension reduction in a way that is appropriate for the specific statistical challenges in microbiome data. The model applies an efficient technique called variational inference.
I'm really enjoying working with both Kevin and Hannah, and I feel very fortunate to have them as my supervisors. Kevin's hard work and dedication to microbiome research is truly inspiring, and it has motivated me to push myself further in this field. The model that we have developed it's called zero-inflated factor analysis, skew normal models. The microbiomes has a lot of challenges such as compositional nature, zero inflation, high dimensionality. We cannot do the usual statistical inference on the microbiomes just as microbiome data, because the sequence in that is not the same as we vary the sample. So we need to do some transformation. So my work was to identify that and incorporate that skewness in the model. And for that, in the factor analysis, we assume that the latent factors are skew normal distribution. I think it's a really great opportunity for me to work on the microbiomes, because this field, it's a sunrising field, and there's a lot of great contribution has been done in the microbiome to study the body, to study the diseases, which is happening because of the disruption in the microbiomes in our body.
In my future research, I plan to touch on a number of different research areas in microbiome analysis: dimension reduction, abundance analysis, and microbial diversity analysis. I plan to develop a cadre of software tools that will allow microbiologists to easily run my models and interpret the results. I have a number of collaborations with microbiologists at the University of Manitoba and Canada-wide, where I can apply my techniques on microbiome data to make a real impact on the research community in the microbiome context.